Welcome to Ibrahim Academia YouTube channel. This is the very first lecture, and this lecture we are going to discuss the physiology of liver, the entire functions of the liver. It is a very broad topic. It is a very huge topic. I can't complete this entire syllabus in just one lecture, so we are going to have a series for this topic, but some functions will be discussed in today's lecture. The functions of liver. Let me give you some solid information. Liver is the largest gland and the largest internal organ of the human body. In the language of science, the liver is called the hepatic organ and the cells of the liver are called hepatocytes. The liver inside the human body is performing more than 500 functions: metabolic functions, the detoxifying functions, the immune functions, the synthesis functions. Liver is doing all these functions, and it counts more than 500. This information is reported in Johns Hopkins Medicine's Research Institute in America. We all know that all the drugs when entered inside the human body, it is detoxified by the liver. But liver is also perform another cycle which is called urea cycle. What is urea cycle? In urea cycle, the liver is detoxifying a chemical which is called NH3. This NH3 comes from the breakdown of the amino acids, the protein metabolism. Ammonia is highly toxic for the neurons. It is very much small. It dissolves readily inside the water. So that's why we have to dilute this molecule. And how much water we need to dilute? Suppose we don't have the urea cycle inside the body. So how much water should we need to dilute the urea? Suppose if there is no urea cycle, so approximately 13 to 14 gram of NH3 will be there in the human body. And how much water it needs? It needs from 80 to 90 liters of water for the dilution to decrease the toxicity of ammonia. And can you drink 90 liters of water in a day? No, this is not possible for the human body. So that's why Allah Almighty has installed a cycle inside the human body which is called the urea cycle. What happens in the urea cycle? This NH3 molecule is converted by the inside the hepatocytes, inside the liver cells, to another less toxic molecule which is called urea. This urea then enters the bloodstream and it reaches the kidneys, and kidneys it is filtered from the blood and excreted in the form of the urine. This is a cycle that liver performs. The urea is made in the liver. Now let's talk about bile. What is this bile? What's inside this gallbladder? There is some water in which there is cholesterol, there is bile salts, bile acids, phospholipids, water, all these electrolytes, etc. All these water, all this the container is installed for the purpose of emulsification. This emulsification will be done inside the intestine. What is emulsification? The breakdown of larger lipid molecules into smaller lipid molecules in order to increase the surface area for the lipases. How this bile is synthesized inside the hepatocyte, inside the liver cells? Primarily, first there is synthesis of bile acid. The bile acid then goes from some processes in which there is the addition of some amino acids, and after the addition of these amino acids, the bile acid is converted into bile salts. This bile salt is then transported through a channel, through a gate, which is called BSEP, bile salt export pump. The bile salt has been exported by this channel, and where this bile salt goes, there is small minute microscopic channels around each hepatocyte which is called canaliculi. The bile salt has been diffused into the canaliculi, and these microscopic channels then drain this bile salt towards the main ducts, and those main ducts finally come to the gallbladder. Another very important function of the liver which is called carbohydrate metabolism. And carbohydrate metabolism, what happens, there is one process which is called glycogenesis. What is glycogenesis and glycogenolysis? Let me give you a very simple example. You have $100 note. On another side you have $1, and the shopkeeper which is living in your neighborhood can't take $100 note. You have to give him $1 to purchase something, to purchase chocolates or other things. So this note, the $100 note, is for the storage purposes, not for direct consumption. You can only consume $1. This $1 is called glucose, and the $100 note is called glycogen. You have to break this $100 note into individual dollars. Then you can consume these notes, these coins. So in liver there is the synthesis of $100 notes which is called glycogenesis, and whenever you feel hungry or there is a flight or fright condition and you need abrupt need of a glycogen, so those $100 notes will be broken down inside the hepatocytes, and this process is called glycogenolysis. Another process there is happening that is called gluconeogenesis. Whenever there is no carbohydrate, to form glucose, so the glucose synthesis occurs from non-carbohydrate substances. For example, the from the fatty acids we can also synthesize the glucose. This process is called gluconeogenesis. All these processes happens inside the hepatocytes, and this comes under the umbrella of carbohydrate metabolism. These were few main functions of the liver. Tomorrow, inshallah, we will discuss the detoxification process inside the liver by taking some relevant example of a tablet, how the tablet has been detoxified inside the hepatocyte, and which enzymes are responsible for this detoxification process. Inshallah, tomorrow we will discuss this.